a while. There you go, recording product. So we're here to talk about the lattice light sheet that we're going to demo in a few weeks, kind of give you an idea of what it is and what it isn't. So the presentation will take about 20 minutes, and then we'll be here for questions afterwards. This is Chase Bryan. He's my 3D application specialist. So we'll be giving the presentation and certainly answer your questions.
Yeah, great.
Right.
Well, hi, everyone. Thank you all for coming. I really appreciate the invitation from the Scissor and the Vanderbilt community at large to come and speak to you today about our lattice light sheet solutions. As Cumberland said, my name is Chase Bryan. I'm the Carl Zeiss 3D imaging specialist for the Southeastern US. He's based in Nashville. I'm based in Atlanta, but I'm not too far from you guys. I just cover a little bit more territory than Cumberland does. Today, I'm going to be talking about the Zeiss lattice light sheet solution. We're really excited to bring this microscope to campus and really see how we can incorporate lattice light sheet technology, or rather Zeiss's interpretation of lattice light sheet technology, into the Vanderbilt workflows, and see what you guys can do with that and the instrumentation that we have to offer, and see how we can apply that for your high impact research that you're doing here at Vandy. I'm aware that you guys have lattice light sheet technology on campus already, so some or maybe all of you are going to be familiar with the principles of lattice light sheet, but I do want to spend a fair amount of time discussing the lattice light sheet technology itself, what that entails, and how Zeiss is approaching lattice light sheet as a modality, an imaging modality. So the initial technique was first published by Eric Betzig and his colleagues in 2014, and has really been credited as starting to usher in a new era in gentle volume imaging. Many of you are probably familiar with spinning disk microscopes. And in 2014, when this first paper was published, spinning disk was really thought as the ideal technology for imaging long-term time-lapse type cell imaging. So when this was published, this really did kind of push forward the idea that there are alternatives to spinning disk. And this paper really does outline how photobleaching and phototoxicity from lattice light sheet imaging are significantly improved or decreased, really, by 1 to 2 orders of magnitude when you're comparing to other 3D imaging approaches like spinning disk. So in this movie here, I've been showing you, this is an example time-lapse movie from a lattice light sheet microscope, a Zeiss lattice light sheet 7, that I think really does highlight some of the benefits of the gentleness of lattice light sheet imaging. So these are some very light sensitive cells, but they were imaged every 5 minutes for over 24 hours. As you can see the time-lapse running in the bottom right here. When we reach the end of the movie, at the end of 24 hours, it's going to jump forward because I actually want to make it move forward. It's not going to cooperate. But at the end of the movie, what we can see is that there are still cells that are nice and happily dividing at the end of the time lapse here after 24 hours of continuous imaging. And we really do think that this wouldn't be possible using any other imaging modality other than lattice light sheet because of the gentleness of the technology and how harsh some of the other imaging modalities can be when you're doing continuous imaging. So lattice light sheet really is able to provide this unprecedented gentleness that helps avoid phototoxicity as much as possible for these long-term time-lapse experiments and long-term volumetric imaging. Imaging with a lattice light sheet can also be performed extremely quickly, so we can capture really fast or dynamic processes as they're happening in our cells, even with 3 dimensions or high spatial resolution, so that we can actually capture really important subcellular details. This movie that I'm showing you here in the top right is an example of a close-up of a cell that's undergoing mitosis. And we're looking at the actin cytoskeleton, the mobility of the actin. And essentially, the imaging that we're doing is not disrupting the dynamic process of mitosis here in these cells as they're dividing. So this here in the center of the screen is maybe a little bit more familiar to some of you, or is at least the initial implementation of a lattice light sheet microscope. I believe this is a photo of the Betzig system at Genelia, or one that's patterned immediately after that type of system. And initial implementations of lattice light sheet looked a lot like this. They're large, they're breadboard-based systems, and they're often or primarily configured as upright microscopes. They're not inverted, and they're not really well suited to live cell imaging, because the objective lenses, because it's an upright system, need to dip into the sample media. And this can cause problems like contamination of your media or contamination of the microscope. And these are things that we're trying to avoid with the Lattice Light Sheet 7 microscope. In addition to the contamination issues, there are limitations around the types of sample carriers and the cover slips that are necessary for imaging on a lattice light sheet in an upright configuration. And these are some of the things we've thought about with our design implementation. So we've actually transformed the lattice light sheet from this system here that you see on the left to a much more user-friendly system that you see here on the right. These are some of the things that we were trying to combat with the implementation to make it much more approachable as a technique. So we've re-engineered the configuration from that upright system into this inverted platform that can handle a wide variety of sample carriers, including multi-well plates and chamber slides. And I'll talk a little bit more about that at the end of the presentation here. But one of the other considerations of this is that there's a lot of manual alignment that has to be done on a breadboard style system. And that was absolutely a consideration in implementing the lattice light sheet as a Zeis solution. All of the necessary system alignments are easily and reproducibly performed in the software. There's no manual turning of screws or adjustments yourself. It's all software managed. And this shares the same familiar Zen interface of all of our other microscopes as well. So the lattice light sheet is going to look familiar to you if you've used another Zeiss microscope previously. So let's talk a little bit about how lattice light sheet imaging actually works. So to understand the principle, I just want to forget about the term lattice for a minute and focus on light sheet. Some of you, or maybe all of you even, have some experience with light sheet microscopy, either with the Zeiss Light Sheet Z1 that's present on campus, or maybe you've had access to one elsewhere. But to illustrate the core principles and advantages of light sheet, I like to use this simple demonstration where the specialized laser pointer isn't generating a point, but rather a beam of light, or a sheet of light. And when this sheet gets scanned over the block here, you can think of this as like a sample in a light sheet. You could see that the resulting optical section gets formed. The resulting optical section is visualized as the laser gets scanned across the sample here. So with a light sheet approach, the illumination is in the entirety of a thin plane that we place in the focal position of the detector. You can see that illustrated here on the left. The illumination comes in at the same focal plane as your detection objective here at the top, meaning we're not exposing these out of focus regions above or below our illumination plane. To better understand the benefits of light sheet microscopy, it's often helpful to compare it to your standard, more traditional imaging techniques like point scanning microscopy or spinning disk. You can see here on the left. With a confocal, by the time you scan across your sample, you're going to build your image pixel by pixel, but doing so means that you're exposing your entire sample to laser, even above or below your focal plane here in the center of the, in this case, the zebrafish, for example. With a light sheet, in comparison, the illumination is focused entirely in this thin plane that's only coinciding with the focal plane of your detection optic, meaning we're not exposing these regions above or below to light. And this highlights two major advantages of light sheet microscopy. One, they're fast, in part because we can use cameras for detection rather than point detectors. And two, light sheets are gentle with substantial reduction in laser exposure, especially for samples that require hundreds or sometimes thousands of Z-planes to capture the entire volume. If you have a large structure, you want something that goes fast and is gentle so you don't bleach your signal as you're acquiring your data. And you can really see the benefits of gentle imaging, for example, with a lattice light sheet quantified here in this graph. So here on a spinning disk confocal, the fluorescence signal is almost entirely lost after 150 seconds of continuous imaging time and 20 acquisitions. However, in comparison, on the right side here, we see the lattice light sheet 7. These same samples displaying a minimal decrease in fluorescence intensity. And this is after almost 300 seconds of acquisition and 3.5 times more acquisitions of the same volume size. So this is 71 volumes over twice as much time, and we're seeing maybe 10% signal loss compared to 90 to 95% signal loss on a spinning disk confocal. Okay, well, this is great, but what's really so special about the lattice light sheet in particular, in comparison to your standard traditional light sheet microscopy? Well, there are a few notable forms of light sheets that have different attributes and benefits to them. The classic light sheet, such as the one seen on the Zeis light sheet Z1, so your classic Gaussian beam light sheet here, is a scanned Gaussian beam. This is ideal for imaging large-scale organisms like things like a zebrafish or Drosophila, or larger samples like thick organoids or cleared pieces of tissue. These are really where you see advantages of the traditional light sheet microscope. However, these really classic Gaussian light sheets are usually too thick to be able to visualize subcellular dynamics. So they're physically too large to be able to see things at a subcellular resolution. So an alternative to your classic Gaussian light sheet is to create a Bessel beam. And this scanned Bessel beam can produce a much thinner middle beam. So we can see this is a much thinner beam here in the center of a Bessel beam, but it also generates a lot of side lobes and generates a lot of out of focus excitation. So even though the light sheet itself is thinner, these outer rings around the Bessel beam will excite out of focus fluorescence and cause additional photo bleaching that you don't necessarily want and is not ideal. So the lattice light sheet really provides the optimal and ideal combination of those first two light sheet options. So when Bessel beams are introduced as a lattice or an overlay of beams, they can cause destructive interference to form between the rings of each Bessel beam. And this results in those outer rings being destructively interfered and leaving only a very long and thin sheet of light at the end. We'll see those side lobes disappear at the very end of that. And this Bessel beam is well suited for gentle volumetric imaging with very low phototoxicity over hours or even days of continuous imaging. So the time lapse that I'm showing you here, I'm not going to let it play because it actually was imaged for 9 hours continuously and is comprised of over 1 million images in this one particular volumetric acquisition. But you can appreciate that there are really fast and really dynamic actin-based ruffles that you can visualize in these cells throughout the duration of this movie. So while this Bessel beam technique results in a lattice structure that's much thinner than that of a normal Gaussian light sheet, one last step is required to create the lattice light sheet necessary for imaging. So the visible lattice pattern needs to be smoothed out, which can then generate your very thin and even light sheet that you need to actually image a sample. This process is called dithering, and it's achieved by very quickly scanning your lattice light sheet to the left and the right. The movie shown below here just shows a quick demonstration of a sample with dithering off and then dithering turned on. The lattice pattern can really obviously be seen on the sample with dithering disabled. But I want to point out that this is something that is automated with a single click in the Zeiss software. It literally is one click to turn dithering on and return your light sheet to this very smooth, continuous form necessary for imaging. So let's take a step back for a minute. And I do want to mention that there are some really key areas that actually do overlap between applications that are a good fit for the lattice light sheet microscope and those samples that are better suited to the larger Gaussian light sheet technology. If you're imaging things on the left side of the spectrum, things like cell monolayers, tissue culture, cellular interactions, these sorts of small scale experiments actually really fit perfectly on the Zeiss lattice light sheet. Larger three-dimensional cell cultures like spheroids and organoids really do kind of hit this middle ground. Depending on how large these things are, these could be a good fit for lattice light sheet, or they might start to trend in the territory of moving to the Gaussian light sheet instead. Smaller embryos, things like Drosophila, developing zebrafish, it's important to consider the overall size of the region of interest. The lattice light sheet can be a very good fit for the smaller organisms seen here. Things like C. elegans also are a very good fit on the lattice light sheet as well. They're really small and really not well suited to a Gaussian light sheet in that case. So the lattice light sheet can be a good fit for early stage embryos, or if you're just interested in things near the surface of an embryo, for example, lattice light sheet would be a good fit for those types of samples. Beyond that, once we start getting into larger samples, things like mouse embryos or cleared tissue, things that are in the millimeter diameter scale, that's when we start to trend towards a better fit on the Gaussian light sheet. So again, larger embryos, cleared tissue really might not be a great fit for lattice light sheet microscopy. But the ability to capture cell volumes in growing samples at speed is the key to why lattice light sheet microscopy is such a powerful and groundbreaking technology. In this movie here, I'm showing you a T cell that's expressing LifeAct fused to GFP. On the left is a color-coded max projection of the entire volume. And we can see the actin ruffles are observed throughout the time series. I haven't studied T cells myself, but my understanding is that these are very motile cells, but they're also very light sensitive. So if we expose them to too much light, they'll actually stop generating these protrusions and these motile dynamic processes. But by using the lattice light sheet 7, we're able to acquire one volume of this particular T cell every 2.5 seconds for the total duration of this time lapse over the course of an hour. So this combination of gentleness and image resolution is not really possible on other imaging modalities. This is going to be unique to the lattice light sheet 7. For a few more examples of some of the specimens we've imaged on the lattice light sheet, here's an example of more T cells, in this case, attacking a cancer cell. So here, the T cells are labeled with cell view maroon. So this is the magenta signal here. And in this experiment, the researchers we worked with were interested in studying the dynamic actin cytoskeleton, not of the T cells in this case, but of the melanoma cell that was being attacked by the T cells. You can imagine this is actually a pretty difficult interaction to visualize with just under standard microscopy techniques. So because we're at that intersection of fast processes and needing large fields of view to see essentially one interaction happening in a dish, this is a great application for the lattice light sheet. One, it's a very gentle modality, and we're able to keep the T cells intact during an imaging session. And two, we want to be able to catch this entire volume as this process of interaction between the cell types occurs. So here we imaged a volume every three minutes for over 2 hours, and we're able to capture this really fascinating data set, watching the T cell interact with the melanoma cell and watch the actin cytoskeleton as it dies after this interaction. So lattice light sheet is not just beneficial for imaging kind of flatter, more two-dimensional cells, but also it's really great at imaging in 3 dimensions and catching really high-resolution volumes. So here we're looking at a 3-dimensional cell culture or an organoid. And this is a really great fit because if you've ever imaged 3-dimensional samples or volumetric imaging on another microscope, what you usually see is a really elongated projection in the Z dimension after you catch your Z stack. This is usually really readily apparent when you look at your cross section in an XZ projection. But here I'm showing you an example of a lattice light sheet image of a spheroid of a U2OS cell organoid with near isotropic resolution in X, Y, and Z. So on the left is actually the XZ projection, which normally would be drastically elongated compared to the XY projection that you see on the right. When I first saw this image, I actually thought the image on the left was my standard XY projection, and that this image on the right was my XZ. But because we're imaging near isotropically, meaning our X, our Y, and our Z dimensions are almost exactly the same on the lattice light sheet, we get a really good high quality volumetric rendering, regardless of the size of our sample here. So not only are we able to improve resolution in time, we're also improving resolution in Z as well. Some of the benefits of the Zeis lattice light sheet really do become extremely important during time-lapse experiments and quantitative analysis, where that isotropic resolution really makes it a lot simpler and easier to do four-dimensional tracking of subcellular structures without disturbing the samples with excessive phototoxicity. So here, using the lattice light sheet 7, we were imaging a early stage dividing C. elegans embryo. And the researchers we were working with were interested in counting and tracking the number of centrosomes, these magenta labeled structures here, during cell divisions, as well as the actual volume of those structures and their sphericity or their shape during this mitotic process over the course of about 2 hours. And thanks to the lattice light sheet, we were able to image continuously for 2 hours and get really robust data that they could quantitate throughout the entire movie. Another great feature of the lattice light sheet 7 is the field of view. So a larger field of view means that you can really easily image much larger volumes, meaning that you're less likely to miss observing a rare event during your imaging session. So the field of view on the lattice light sheet 7 is about 300 microns in Y, but in the X dimension, you can scan essentially an entire multi-well plate. So the sample carrier is your limit on the field of view that you can acquire here on the lattice light sheet 7. In this example, the researchers were looking at lamin B fused to GFP, localizing to the nuclear envelope during cell division in these iPSCs. Since it was impossible to predict at the start of the experiment which of their cells was going to divide, it really is ideal to have a much larger field of view where you can actually image dozens to hundreds of cells at a time and really give yourself the best shot to visualize the most cells to give you a higher probability of seeing the event you want to see. This data set encompassed 300 by 100 by 30 microns, so actually a fairly large volume to consider that we were imaging once per minute for 500 time points, or just over 8 hours. The exceptional stability of the system, combined with this large field of view, means that many of the mitotic events that were captured over the course of this 8 hours wouldn't have been possible with another system that was less stable or had a smaller field of view. So having a microscope with high technical capability, but that requires a dedicated technician to run it, does work well for some users, but often it's not the best. It's really not the best solution for having a wide user base and achieving the throughput of experiments that are expected from a central piece of equipment in a core imaging facility. That's why the Zeis lattice light sheet 7 has really been designed and engineered to make sure that you can simply put your sample on the stage from the top. You can close your sample chamber like you saw just a few moments ago. And then with just a few mouse clicks in the software, you can get a transmitted light preview of your sample and then quickly transition into fluorescence imaging as quickly as you watched in this movie. The simplicity of this workflow means that even inexperienced users will be confident using this microscope and really minimizes the amount of time required for training and maintenance and maximizes the time that your scientists can actually acquire meaningful data on the system. Oh, PowerPoint did that thing where it crashed. Give me just a moment to bring that back up. Sorry, folks. One downside to using a Microsoft product is you know it's going to crash when you don't want it to.
I think it was on the email that was sent out, but the demo weeks are from the 11th, 29th of September. So that'll make the sign up and slide.
Yep. Okay, sorry about that. We're back. So the Lattice Light Sheet 7 has an integrated incubation and auto immersion solution such that you can run the instrument after it's configured and leave it to run for days at a time. The stability in incubation is really complemented by the 5-axis stage control that you see in this movie down here. 5-axis meaning it's adjustable in X, Y, Z, and tilt, both in X and Y. This stage is what really simplifies the alignment of the system and makes it a much easier and really approachable solution for lattice light sheet microscopy. This is what enables us to really have it be such a time saver and be such a robust and friendly system for users to put their samples on. As you saw in the last couple of slides, the geometry of the Lattice Light Sheet 7 is inverted compared to upright. So this familiar configuration ensures that you have full sample flexibility, and it's compatible with all standard sample carriers, meaning if you're doing live imaging, there's a good chance that you can just move your sample directly onto the Lattice Light Sheet. Anything that you are already using, things like glass bottom Petri dishes, multi-well plates, chamber slides, these are already going to be compatible with the Lattice Light Sheet 7. So anything that's currently being imaged on another microscope, such as a spinning disc or a laser scanning confocal, there's a really good chance you can immediately port that experiment over to the Lattice Light Sheet 7. I'll talk about specifics in a minute or two, but the laser lines are also those commonly used for live imaging. And that's an important consideration in making sure that you can just bring your samples directly to this system. You probably noticed in the last slide, the objectives are positioned below the sample. The illumination objective here on the left and the detection optic on the right are precisely positioned and high numerical aperture objectives that give us high resolution. So these objectives are positioned 90 degrees away from each other, such that the illumination is perfectly aligned between the illumination objective and your detection optic. And it also dictates that your samples get imaged at an angle. I'll talk a little bit more about that in a slide or two. But just briefly, I do want to talk about one or two more of the really impressive innovations that went into designing the core optics of this system with the goal of stability and image quality that allow us to compensate for refractive index changes with your sample and give us the best image quality possible. So the first one is the meniscus lens. This is in place to correct for the air-water interface that we see here. And this allows us to use water immersion, which is important because the vast majority of our live imaging experiments are going to be done in water immersion. We need to match that refractive index. So this is a really key innovation, this meniscus lens itself, because one, it allows for thermal isolation of our sample, and it also improves stability and refractive index matching for the optics of this system. The other components of these optics are the adaptive optics that we see here in the detection objective. These are what are called freeform optics, and these two plates actually move and can be adjusted to compensate for cover glass thickness or small refractive index changes to really give us a really high quality image. And these are adjustments that we can make on the fly in the software. There's nothing that we have to really dive into the guts of the scope to change. It's all really easy to manipulate yourself. Exactly. So it's not necessarily going to be a very I don't want to say reproducible because it's absolutely reproducible, but it is look at it, find your best image, and then go with it. You don't have to take calipers to your cover slip and figure out how thick it is. You just change a slider, see where your image is really coming through the crispest, and then move on. It's really quite simple to implement. I mentioned a moment ago that your sample is getting imaged at an angle due to the geometry of the objectives. And on the left, you can see what the sample actually looks like to the cameras of the lattice light sheet. So this is what your sample looks like as the data is being acquired. We see a nice thin section of our specimen on the cover glass. But what you see on the right is more in line with what you expect from any other microscope. And the workflows in the Zen software for the lattice light sheet make it very easy to convert this image the camera sees into a transformed de-skewed image like you would expect to see on any other microscope that you see here on the right. One of the last things that I want to talk about is that there are no eyepieces on the system. You probably already saw that. There's clearly no way to see your sample by looking at the system itself. And this is because we have ways in the software to navigate around without the eyepieces. So in this example, we're going to start with this diagram of a four chamber chamber slide. And we immediately can see that there's an overview image that was taken with transmitted light. And if we have larger samples in the chamber, like organoids or an embryo, you could see that with this transmitted light view. But instead, what we're looking at is fluorescent cells on a cover slip. So we can actually, instead of using the transmitted light to visualize those, just do a quick fluorescence preview scan of a small region of our cover slip. And then we can actually see the cells themselves that we're interested in performing a time lapse on. So it's very easy to navigate around using the lattice light sheet system and the Zen software, even if you don't have the eyepieces to look through and find your area of interest yourself. We've made it really simple in the software to do it instead. Before we wrap things up, I just want to show you one more example that illustrates how easy it is to access lattice light sheet technology and how it generates more meaningful results and more meaningful data that can accelerate your research. This data set here shows the dynamics of kinetochores during mitosis. So the powerful combination of the gentle volumetric imaging that you can expect on a lattice light sheet 7 with the near isotropic subcellular resolution of this system gives you the high quality images that you need to perform high quality and powerful analysis, like you can with Zeis software, like Ariva's Vision 4D, where you could take this to other three-dimensional rendering software as well and perform these high power analyses on really high time resolution time lapses. Oops. The spatial resolution of this system is going to be comparable to a confocal. It's around 300 nanometers in X, Y, and Z. So that's where the advantage really comes in, is with that Z resolution versus a system where you're imaging through and then detecting through the same objective. We get the isotropic resolution on this system. So with that, I think I've highlighted a number of the sample types that we can image on the lattice light sheet 7 and the workflows that we can start to work with on this type of microscope. We obviously didn't have time to show you all of these examples today, but we do have researchers using the lattice light sheet 7 to image things like bacteria, yeast, small animal oocytes. Anything on this list, we are more than happy to put on the lattice light sheet and see how it performs for these types of samples. The lattice light sheet 7, I think, really does provide the best solution for very fast volumetric imaging. It's very gentle. With minimal phototoxicity, you can perform time lapses for hours or up to days of continuous imaging. It provides near isotropic confocal-like resolution, as we just mentioned, the resolution capabilities of the system. And it is very simple in that it is a standard inverted configuration that can accommodate your standard sample carriers. It's a small footprint, it's easy to maintain, and it's easy to use. And I think all of these come together to make it a very powerful system for a core facility like you folks have here at Vanderbilt. So let's talk about sample carriers. If you're interested in imaging on this system, we have several different stage inserts for the Lattice Light Sheet 7. I'll send this out to the group at the end of this, but we have stage inserts that are compatible with 35 mil dishes, slides, chamber slides, multi-well plates. On a Lattice Light Sheet 7, the working distance should be as close to the cover glass as possible. We can image really effectively up to 200 microns, depending on the thickness and the opacity of your sample, maybe a little more, maybe a little less. The system comes equipped with 3 laser lines, a 488 for your greens, 561s for your reds, and 640 for your far reds. Notably, there is no UV laser on the Lattice Light Sheet 7. This is a live imaging system, and UV will really push your live imaging into dead imaging really fast. So we don't put a 405 laser on this microscope. But if you have vital dyes that are necessary in the UV or the blue range, chat with us. We have far red dyes or green dyes we can recommend as well. And then finally, the aqueous media is really the best fit for this system because it's a water immersion system. And we prefer phenol red free media when possible to try and minimize any kind of background or autofluorescence, if you can.
How is this handled?
So great question. This is a 2-camera system. So we have a filter in place that will split the light. We actually have several filters for options for splitting the light. So we can acquire 2 channels simultaneously, 3 channels if we do a sequential split. So you can do multi-channel acquisition on this system, either simultaneously with 2 or sequentially for 3. This is the last slide that I have to show. How fast does it scan? So obviously that'll depend on the volume you need to acquire, but we do have data showing that I don't actually have the millimeter spec, but we can capture volumes in under a second, depending on your size of field of view. So we can do repetitive volumetric imaging, 1 hertz plus, depending on the size of your sample. Okay.
So to turn that question a little bit different.
Mm-hmm.
Let's say I have a very bright fluorescent line.
Mm-hmm.
What's the fastest frame that is?
You can drive the system. I think the cameras are compatible with 1 millisecond exposure times. They might be software limited to 5 milliseconds, but we're in the single millisecond range. I would need to verify that, but I believe that you can drive it at 1 millisecond frame times. So coupling that with, just translating your stage across, you could get 1,000 FPS on this system. So the system that will be on campus is equipped with 2 Hamamatsu fusions. So these are really sensitive, high quantum efficiency cameras. These are state-of-the-art Hamamatsus. CMOS, I believe. Yes, SCMOS. Correct me if I'm wrong on that, Cumberland. I think they're CMOS. These are not the back thin fusions, no.
So you mentioned voxel resampling happens on the fly.
We can, so there's a direct processing pipeline that we can send your data through to do the voxel resampling as the data comes through, or you can reprocess the data at the end and deconvolve if you want to. Yes, yeah, you can deskew, you can deconvolve. Deconvolution does take more time, obviously, because there's There's a lot of data that this system can generate, and it will take time to reconstruct that data. But yeah, we can do this on the fly or as a post-processing step as well.
So I take it you need fairly bright structures to even do that, right? Because everything that you show is like a major brightest things in the south.
Right. No, the examples that we show, they're a little bit cherry picked. They look really nice because they're really bright. But that's not to say that this system can only work with bright samples, especially having moved to the Hamamatsu Fusions. This is actually the second generation of camera that we've implemented on this system. These are much more sensitive than the previous generation of camera we were using, and they are very friendly with dim samples. So maybe a little more than 1 millisecond exposure times, but you can still get really good, really high signal to noise on dim samples using these cameras with the lasers on the system.
So the Fusion is better than Orca Flash.
The Fusion is kind of, my understanding is it's like the next in line on the Orca Flash line. Yeah, so these are definitely state-of-the-art, highest end cameras we could equip the system with.
The best thing they can be.
I think it's 8.6. You need to go to 92.
Yeah, I believe the back thin would be 92. Anecdotally, the back thin has a higher signal to noise, or has a higher noise floor. So I think this actually does give the better image quality, not being back thinned, but sacrificing a little bit on the QE side of things.
Better dynamic.
Yes, I believe so.
Is there the possibility of switching out the camera? Let's say I have a very low signal.
Mm-hmm.
Could I put an EMCCD in there?
As it's configured right now, we're only offering the lattice light sheet with the CMOS cameras, but I wouldn't say never, just that it's not as it's configured currently. And that's not what you'll see on campus in a couple of weeks.
Well, that's the obvious choice.
Yeah.
Yeah.
But yeah, I totally understand the desire for a CCD option as well. Okay, I did see we had one question in the chat. Oh, no, that was just Jenny saying, put questions in the chat. Okay, great. If anybody has any other questions, please, Cumberland and I are going to stay around for a little while. We could chat afterwards. Oh, yeah, please, go ahead.
Well, let's say we want to do like some kind of a treatment experiment. How accessible is the sample as we go?
That's a great question. So the sample chamber is enclosed during the acquisition, but there are injection ports, so we can route tubing into the sample chamber. You can do an infusion on the fly during the experiment, and long term, I believe that there is triggering capability on the system. So if you wanted to run something like an infusion pump coupled with the triggering in the software, I believe that would be a possibility as well. So it's accessible in that you can run the system without the lid, or you can run a tube into the sample chamber and have it be a heated chamber, and triggering should be an option as well.
So you can add things, but you cannot say change the or something off. Probably you need to flow through.
You would need to implement a secondary flow through type of setup yourself. Yeah, that's not necessarily baked into the workflow itself, but that is something you could implement as part of this workflow. Go ahead.
Temperature and gas control are coming.
Yes, absolutely. It's integrated incubation. So that's part of the system, is to have it incubated and humidified. So yes, CO2 and temperature control are integrated into the system by default. Okay, thank you guys for coming. It's been a pleasure to speak to the Vanderbilt community, and we're really excited to have the system on campus. I didn't have the dates listed here, but we will have the system on campus from September 11th through September 29th. So please reach out to us if you're interested in imaging during that time frame. Thank you very much.
Thank you, everyone.